Hello, this is Mary with Noteworthy Science. Today we're going to be going over subtopic B3.2 Transport at the additional higher level. Under the curriculum matrix, this falls under theme B, form and function, and under the level of organization of 3, organisms. Questions to keep in the back of your mind for this subtopic is really, 1, what adaptations are there to transport fluids in both animals and plants, and how does this process differ between animals and plants? At the additional higher level, there are 8 learning outcomes where we'll be going over this process in significantly more depth. Let's get started. The HL component of this subtopic is genuinely so interesting because we get to actually go into the anatomy of the heart, the mammalian heart. We get to look at the circulation. We get to look at how does pumping actually work, something called the cardiac cycle. We get to delve into what's actually happening at the tissue level during exchanges of nutrients, and we get to dive more deep into what's going on during translocation in plants. So there's a lot of extra information that I think it's kind of a shame that SL doesn't have this. I think this is really interesting in terms of content. So the first thing that we're going to talk about is different ways in which blood can circulate in different organisms. And so the simplest way that we have is something called single circulation. And this type of circulation is one that you find in bony fish. There's a really good simulation that if you go to the resources page, and it's this website right here, and it shows a really good understanding as to how different circulations work in different animals. So the key here is just, these are what the heart icons mean. These are the different tissues. As I mentioned before, red represents oxygenated blood, blue represents deoxygenated blood, and purple here may confuse you, but this is actually mixed blood. It's both oxygenated and deoxygenated. And that's only true in some organisms. So let's start off with just fish. We have the heart, and then notice deoxygenated blood is leading to the heart. As it's moving up through the gills, it's now becoming oxygenated. That's now circulating the head, the body tissues, going around, then it's going through the kidneys, and then now it's become deoxygenated and it's back to the heart again. So this system, as you guys notice here, this would be called a single circulation because it's only passing through the heart once per cycle. So it's passing through, becoming oxygenated in the gills, sent to the whole body, and then back to the heart again. So if we were to zoom into just the heart, notice that there's 2 chambers. So we have the atrium and we have the ventricle. So as deoxygenated blood comes in, it enters into the atrium pumps it into the ventricle, and then the ventricle pumps it into the rest of the arterioles to then go to the gills in order to become oxygenated. But notice that the entire heart in this section would only contain deoxygenated blood because it's returned from the tissues, deoxygenated, and then it's being sent to the gills to become oxygenated. What do you notice about the difference in tissue between the atrium and ventricle if this represents muscle tissue? And give me a reason for that difference. The atrium only has to pump blood really from here to here. So it's not that difficult to do, but this pump has to make it not just to the gills, but to the rest of the whole body. So this has to be a much stronger push. And actually this will be a recurring theme even when you look at the tissues in the mammalian heart as well, where you see that the muscles in the atrium are going to be significantly smaller than the muscles in the ventricles because the ventricles are pushing blood much further distance than the atrium are. Right? Then here is my 2-chambered heart. Here would be the atrium, here would be the ventricle. And again, I'm using the same convention here where I'm showing blue for deoxygenated blood and red for oxygenated blood. And now, if I start, let's just say here, I would have my atrium pumped to the ventricle, pump to the artery. All arteries pump blood away from the heart. And again, look at this as an example. If you were under the impression that artery means oxygenated blood, this is deoxygenated. So that's not true. Artery just means blood away from the heart. So blood is now moving towards the gills in order to become oxygenated, right? And this is where gas exchange happens. We've now offloaded that carbon dioxide. We've now taken in oxygen. This is now being sent to the rest of the body. And then that would be like the tissues of the body. So this would be all over the fish. And that would ultimately, gas exchange would happen here, oxygen delivered, CO2 taken in, and now this returns to the heart again. So this is where the term of single circulation happens, because it's just one circuit all the way around. Now this is not true for mammalian hearts. So mammalian hearts have what's called double circulation. Here is an animation of a mammalian circulation, and notice that it looks definitely a little bit more complicated than it did in the fish, right? So take a look at what's going on here. Notice that before, the heart in the fish, it was only deoxygenated blood passing through there. But in the mammalian heart, you have both oxygenated and deoxygenated blood passing here, and there's no mixing. So one side of the heart is only oxygenated blood, one side of the heart is only deoxygenated blood. Now the deoxygenated blood, again, in the fish, we sent the deoxygenated blood to be oxygenated into the gills. In mammals, we're going to send that to the lungs to become oxygenated, and then that returns back to the heart. But notice it's returning to the other side of the heart to then pump that to the rest of the body. So there's actually a double circulation path. So let's zoom into the heart here. Notice that in the mammalian heart, there's actually 4 chambers. 1, 2, 3, 4. Now, we're going to use the same naming conventions that we did with fish. The chambers at the top were called atria, or 1 atrium, 2 atria, and then the bottom were the ventricles. Now again, look at the differences in terms of tissue, right? We have much thinner, much thicker, and notice that the blood is actually partitioned right here. So we have oxygenated blood, deoxygenated blood. So this would be sent to the lungs, this would be sent to the rest of the body, of the aorta. So this is the double circulatory path. Now as a extension here, take a look at the frog. It's actually quite interesting. And I want to do a zoom in on the heart. Now look at the difference between the heart here. There's mixing here. So they still have 2 atria, but a single ventricle. So this means that oxygenated and deoxygenated blood are mixing. Now that is significantly less efficient. Can anyone think of a reason why amphibians, like a frog, are able to have this less efficient heart? They can also perform gas exchange via their skin. So they're not reliant only on gas exchange in the lungs, they can also do gas exchange through the skin. So this is definitely less efficient, but they can kind of get away with it, so to speak, because they have that extra adaptation that we do not have. Incidentally, when we do ecology, you'll notice that amphibians, because of this, are very, very vulnerable to air pollution and to water pollution. Because they do so much of their exchange via their skin, they're very, very sensitive to changes that can occur. And in fact, if you look at the list of critical endangered species around the world, amphibians are a huge number of those animals. Because anywhere where you have air pollution or water pollution, they're very, very vulnerable to these changes. Again, this is not in the curriculum, but I just thought it would be quite interesting to take a look at the differences here. A few things about the mammalian heart before we go through, and I think this is a massive area of confusion, and I think it's understandable. When you're looking at this diagram, when you're looking at a picture of a heart or a diagram of a heart, the way that you distinguish left from right is very important. Now, when I'm looking at this, if I'm looking at this straight on, I would say, oh, this is right and left. No. The way that we label anatomy is as if this is in my body. So this is the left, this is the right. So this is the left-hand side, this is the right-hand side. Keep that in the back of your mind because this is a common source of confusion on tests. It's a common source of misunderstandings. And so you have to imagine organs as if they're in you, if that makes sense. So we have the right atrium. The right atrium has collected deoxygenated blood from the whole body. That gets pumped to the right ventricle. That right ventricle is then going to pump blood where? Well, to the lungs in order to become oxygenated. This is where gas exchange is going to happen, offloading of CO2 onloading of O2. That gets sent back to the now left atrium. The left atrium then pumps blood to the left ventricle. And then from the left ventricle, we pump blood to the rest of the body. And then the tissues perform gas exchange, and that returns back to the right-hand side of the heart. So for this reason, we call this path double circulation, because you have both the what's called the pulmonary circuit, or the circuit that involves the lungs, and you have the systemic circuit, or the circuit that involves the body. Both the pulmonary and systemic circuit are happening simultaneously. Do not think like it's first going to the lungs, then the rest of the body. They're happening at the same time, right? So I know when we often describe the flow of blood through the heart into the body, we're often going to use like a step-by-step manner, but just keep in the back of your mind that this is happening simultaneously, right? We are going to be able to label all of this. All of this will make sense, I promise. I know right now it looks super complicated. It's not. We're gonna work through this. So I'm gonna start explaining this path as if the deoxygenated blood is going to the heart. So that means all your deoxygenated blood is gonna be collected from both the top part of your body and the bottom part of your body. Now, what kind of vessels bring blood to the heart? Veins. Now, the two largest veins in your body are what's called the superior vena cava and the inferior vena cava. The superior means it's collecting all the deoxygenated blood from the top half of your body, basically, and the inferior vena cava is collecting deoxygenated blood from kind of the lower half of your body. Both of these are collected into that first chamber that we just looked at. And because this is deoxygenated blood, take a look at the heart, put this in my body. This is the right-hand side, which means that it's now collecting into the right atrium, right? So we have superior, inferior, collecting into the right atrium. Now the right atrium is now going to pump blood to the right ventricle. But notice that there's something in the way here. That zoomed in is something that looks like this. It's a valve, and we know valves even when we looked at the structure of veins, that they allow one-way flow. In this case, I only want blood moving in this direction, not backwards. And the name comes from tricuspid, tri for three, and a cusp is almost like the three parts of the valve. So I tried to draw it here. If you take a look at the valve, there's 1, 2, 3 cusps that make it up. And that's what causes them to open, close, shut. And AV, atrioventricular, which is a fancy way of saying the AV valve, going here. Okay, so we've collected blood, pumped it into the right ventricle. Now the right ventricle is now going to send blood to the lungs. Now what word did we use to describe something referring to the lungs? Pulmonary. So, and what vessel moves blood away from the heart? An artery. So this would be called the pulmonary arteries. Now notice, this is a really weird exception here. Pulmonary arteries are carrying deoxygenated blood because they're transferring it to the lungs to become oxygenated. So you have, of course, the left and the right pulmonary artery because that's sending it to the left lungs and the right lung. Now, in the lungs, gas exchange occurs. We already talked about that in B3.1. Now, blood is now going to be coming back to the heart. So again, what words are we going to use to describe that? We have pulmonary, and vessels that go towards the heart are veins. And again, this is a weird exception here where pulmonary veins carry oxygenated blood back to the heart. And as we looked at in our animation, that's now going to collect now on the left-hand side of the heart in the left atrium. Now we're in the left atrium. From the left atrium, we're now going to pump blood into the left ventricle. And the same word that we're going to use here, this is also an AV valve, but notice the difference in anatomy here. It actually is composed of 2 cusps. So it's called a bicuspid AV valve. I'm including mitral because depending on where you look online, it may also be called this. I don't think you have to use this, but you may see the term mitral valve, and that's what they're referring to. This goes by many names. So we have left atrium through the bicuspid AV valve now into the left ventricle. And now the left ventricle is then going to pump blood through the largest artery into the whole body, and that being the aorta. And that's going to send blood everywhere. So when we are taking our pulse, our carotid pulse, we were measuring it here. You can feel it very strongly because that's coming directly from the aorta up into the neck. And there's plenty of blood flow to your brain. This is in fact where you're going to have the highest blood pressure, is right here. Now, notice I've so far only talked about 2 different valves. There are 2 other valves. And these are the valves that are leading to the pulmonary arteries and to the aorta. Because again, I don't want blood flowing backwards. Now, these valves have a different name, and these names come from, again, describing their shape. Semilunar, what does that mean? Half a moon. So semi means half, lunar means moon, so they have like a crescent shape, if that makes sense. So that's where their name comes from. So they're semilunar valves. And how can I distinguish this semilunar valve from this semilunar valve? Well, I'm going to describe it by which vessel they're going to. So this would be the pulmonary semilunar valve. This would be the aortic semilunar valve right here. Okay. Now, a few other things that I want to keep in mind. Unlike amphibians, we have 4 chambers. 1, 2, 3, 4. So this structure right here ensures that our blood does not mix. That structure is called the septum. In fact, there's even an unfortunate sometimes birth defect or a condition where this has not closed properly, and there'd have to be surgery to rectify that, because you can understand why this could be very problematic now. What do you notice between the relative size of the muscles in the left and the right ventricle? And again, provide an explanation as to why that difference is there. So the left ventricle has to pump blood to the entire body. The right ventricle has to pump blood to the lungs. That's a much shorter path. So there's less actual cardiac muscle here than there was here. Now, as we've mentioned back when I think we did cell specialization, we looked at cardiac muscle before. What are some adaptations of cardiac muscle? If you take a zoom in on my image here, the little discs that I drew here are called intercalated discs. And what that means is that an electrical impulse is able to travel through this simultaneously. Unlike skeletal muscles, these are individual cells, right? Skeletal muscles were multinucleated. These are not multinucleated. This is one cell, one cell, one cell, one cell. But they're joined through these intercalated discs, allowing very efficient and simultaneous propagation of what's called an action potential. So it means that when this contracts, they all contract in a wave. So we've now have hopefully an understanding as to the names of what all these things do, but as something that I'm sure you're already maybe tired of me saying, how is the structure related to its function, i.e. what adaptations is going on here that allows a very efficient pumping of blood? So that's what this bottom part is, which is how is the mammalian heart, or the heart within mammals, adapted to basically deliver this pressurized blood to the arteries and flow through the body. So as we just talked about, cardiac muscle is a very special kind of tissue because the signal that causes your cardiac muscles to contract does not originate from the brain. It originates from the heart itself. So this is where the term myogenic comes from. Myo means muscle. So it originates from the muscles themselves. So there are going to be cells that we're going to talk about in a second called pacemaker cells. And these cells actually are the ones that are initiating contraction. This is different from every other muscle. All other muscles receive their direction, so to speak, from the brain. This is not true. These originated from the heart itself. There's going to be a section in your heart that we're going to talk about in a second called the pacemaker cells, or specifically the sinoatrial node. If here is my simplified version of my heart, right atrium, right ventricle, left ventricle, the SA or sinoatrial node is up here, and then you have another node called the AV node. These are your pacemaker cells right here. This is what's originating all of the contraction in the heart up here. The idea that the signal is originating in the heart, we have a term for that. We can say that these cells are autorhythmic, meaning that they are sending their own rhythm. The rhythm is not coming from elsewhere. And so that's what's initiating these heart contractions. It's almost like if the term pacemaker was what I think about is if you've ever seen like rowing, like on professional rowing teams, and you see that there's an individual in the boat that's actually not rowing, their main job is to set the pace. That's, I think, kind of what I think about with these cells. They're the ones that are setting, go now, go now, go now, go now. Now, as we'll talk about in another topic, these pacemakers can be modified by the brain. So someone can be like, hey, go faster. And he's like, okay. But they're still the ones originating it, but it can be either increased or slowed down by the brain. So it can, or hormones. It can be modified. The originating signal is always going to be coming from the heart itself. Now, these chambers, we said that we're a 4-chambered heart. We said we have atria and ventricles. How are they adapted to their function? Well, these atria, these are going to be your upper chambers. They're always going to be collecting blood at low pressure, right? So because they're always being kind of delivered by veins. So that means that if something's receiving blood at relatively low pressure, then we know they don't have to be like super beefy and have a lot of things for high pressure because they don't need to, really. So that means that their muscular walls are typically much thinner, as you guys already deduced from looking at different diagrams. And their main role really is just to squeeze or contract to pump blood into the lower chambers or the ventricles. Now the ventricles, as you guys already deduced, these are the lower chambers of the heart, and they collect blood from the atria, but they're going to have much thicker muscular walls because they have to send blood a much larger distance away. So they have to be adapted to pumping at very high pressure, and thus you can identify a ventricle from an atria very easily by the relative thickness of their muscular walls. Now the valves will close to prevent backflow. And what I mean by backflow is I don't want blood flowing back in this direction. So the main role of valves, generally speaking, whether they're in your veins or whether they're in your heart, is to allow one-way flow. That's the point. So they're only going to allow fluid to move in they should allow fluid to only move in a single direction. And so the AV valves, of course, are going to prevent backflow to the atria. We only want blood flowing from atria to ventricles. We do not want any blood flowing from ventricles to atria. And so the way that valves work, something that we're going to explore a lot in the next learning outcome, is it all has to do with a pressure differential, right? If you have a higher pressure on one side and a lower pressure on the other, that's what's going to cause the valves to either be open or closed. So they're going to be open if the atrial pressure is higher than the ventricular pressure. So how can I make this pressure higher than this pressure? Decrease in volume, hence it's contracting. So when this contracts, the pressure has now increased, causing this pressure to be greater than this pressure. That causes this to open and blood flows in. So it's really all the pressure differential. So the heart can manipulate blood flow by contracting or relaxing. If I'm contracting something, I'm going to increase the relative pressure in that area. If I'm relaxing, I'm going to decrease the relative pressure in that area. So that means that when the atria are contracted, the ventricles must be relaxed, right? So these would contract simultaneously, high pressure, blood flows down into here. That would also mean then, when are the AV valves closed? If I want these to slam shut, then I need this pressure to be higher than this pressure. And again, how do I do that? Contraction. When the ventricles contract, this pressure becomes higher, these slam shut. Let's consider the pressure differentials in the semilunar valves. Deduce when the semilunar valves will be open and when will the semilunar valves be closed. So once again, think of all about valves opening and closing by pressure differentials. If this is now contracting, as we said, pressure here will be very high. That's going to be higher than this pressure. That's going to cause these to close. But this pressure is going to be higher than arterial pressure, or pressure in the arteries. So that means these will open, causing blood to flow in. As more and more blood enters the arteries, that's going to increase arterial pressure, and at some point these will relax as well, causing these to now shut to prevent blood from flowing back into the ventricles. So it's a really cool system here where you have these 4 chambers with these 4 valves, each ensuring that blood is only flowing in one direction: atria to ventricles to arteries. Atria to ventricles to arteries. So it's always going to flow in that same way. And actually, whether it's double or single circulation, it's the same. Look at the fish. Veins, atrium, ventricle, arteries. Veins, atria, ventricle, arteries. Always in that same path every single time. So this is the atrium up here. So this is all ventricle. So atria are up here. Atria, ventricles out. Atria, ventricles out. In that way. So when you look at something like this large part down here, these are the ventricles. And actually I always remember it that way because they kind of make a V. These are the ventricles and the atria are up here. And notice that this is the valve, this is the AV valve, so that's bringing blood into here. And then here are the arteries, and that's the valve leading, that's the semilunar valve leading to the arteries right here. Look directly down and you can see here are 2 valves, here are 2 valves. So these are the AV valves, these are the semilunar valves. So when you really look at it from this perspective, I think it gives you a better understanding as to where all of this is relative to each other. We're now going to go into the cardiac cycle, which is essentially the sequence of events that's actually going on that allows blood to be pumped efficiently through the heart. And this is going to be broken down in a few stages. The very first thing that we're going to talk about is what's initiating this contraction in the first place. And that's going to be this node right here called the sinoatrial node, or the SA node for short. Now the SA node is basically a collection of myogenic pacemaker cells. And these are special kind of cardiac muscle cells that are able to initiate their own action potential. So they, not the brain, are the initiator of this action potential wave. In order to have efficient blood flow, the atria have to contract before the ventricles do, and they need to contract in unison. So how is this accomplished? If we just had a wave pass this way, then it would cause the entire heart to beat in one big wave, and we don't necessarily want that because the blood has to flow down and then back up again. So how do we do this? Well, the SA node is going to send a depolarization wave throughout both the right and the left atria first. This signal causes the cardiac muscle cells in the atria to contract, and this is what's going to cause blood to flow down into the ventricles. Now, before the ventricles contract, all of this is going to be gathered in a secondary node called the AV node. The AV node is what's located on the wall or around the junction between the atrium and the ventricle. The signal then gathers here, and that causes a slight delay, but like a 0.1 second delay. That's key because that ensures all the atria contract first and then the ventricles contract. This depolarization wave, however, is not going to go just down this way. Instead, the depolarization wave is going to flow in this direction. The question is why? Why is the action potential sent down the septum and then outwards around through these fibers called Purkinje fibers. Explain the reason for this. If the wave actually was propagated just downwards this way, then what would happen is that the ventricles would contract from top to bottom. Well, what would that mean for the blood? It would mean the blood is going to be pushed towards the bottom part, which is not what we want. We want the blood to actually flow up into the arteries, both the pulmonary arteries and the aorta. So there's a really clever solution to this. And by first getting this to come down, these fibers in the center, they then go out through the Purkinje fibers. And the Purkinje fibers are what are actually innervating the cardiac muscles within the ventricles. And so this causes the ventricles to actually contract from bottom to top. And this squeezes blood upwards into the actual semilunar valves towards the arteries. So first the atria contract in unison, and then the ventricles contract in unison from bottom to top, forcing blood out. Anytime that we're referring to the atria or the ventricles contracting, we're going to call that systole. So if the atria are contracting, that would be atrial systole. If the ventricles are contracting, that's called ventricular systole. If a part of the heart is not contracting or relaxing, then we would say it's in diastole. So let's now go through what's really causing blood to go from atria to ventricles and then ventricles into arteries. For this to occur, they have to pass through valves. Now, valves, as we discussed, allow one-way flow of blood. And the way that valves work is really all to do with a pressure differential. If the pressure is greater on one side than the other, then that valve will open up, causing blood to flow in that direction. But if pressure now over here is greater than this one, it's now going to cause the valve to slam back shut. So in this way, depending on the valve, if pressure is higher on one side, it's going to allow it to open. But if pressure is higher on the opposite side, it causes it to close. So let's look at how this works in both atrial systole and ventricular systole. And the best way to really look at this is through this kind of graph. A learning outcome is for us to be able to interpret such a graph. Now this graph looks quite complex, and that's just because I tried to include everything in one big graph, but I'm going to help you make sense of this. So on the x-axis is just time, but notice how small the increments of time are. These increments are in 0.1 second. So this cardiac cycle that takes place is less than 1 second. That's kind of remarkable. So this whole thing is happening fairly quickly. On the y-axis is pressure, and pressure is going to be measured in millimeters of mercury here. Now, I have drawn 3 different curves that are color-coded. And each of these curves represents change in pressure over time throughout the cardiac cycle. So the curve in red represents the pressure differences happening in the aorta, the really big artery leading out of the heart. The curve in orange here represents the pressure differences happening on the left ventricle. And the curve in purple here represents pressure differences happening in the left atrium. Now, keep in mind that these curves would look very similar if we were to look at the right ventricle and atrium. They may be lower levels because the pressure differences are typically smaller in the right than they are in the left, but we're only going to focus on the left hand here. So let's begin in our first part of our cycle here, which would be atrial systole and of course ventricular diastole. Notice when one of these chambers are in systole, the other must be in diastole and vice versa, because if both were in systole, both would be contracting simultaneously. That's not going to happen. Now, both can be in diastole, where the heart is going to be passively filling, but both cannot be in systole simultaneously. So let's begin with first atrial systole. As we mentioned, the SA node is going to propagate a signal across the left and right atrium, causing the atria to start to contract. Now, if the atria are starting to contract, that means that the space is going to get smaller, which means that pressure is going to increase. So as the atria are contracting, Pressure is slowly rising here. Now, how is it that you don't see a really big difference in pressure? Well, remember that the atria are actually quite small. There's not that much pressure differences here, and they are filling from veins coming in. So the pressure differences aren't going to be super large, but there is going to be a rise. Now, as blood flows from the atria into the ventricles are then going to start to contract. That's when the signal gets propagated around the Purkinje fibers and the ventricles begin to contract. Once the ventricles begin to contract, that's going to rapidly increase blood pressure within the ventricles. Once the pressure in the ventricles gets larger than the pressure in the atria, the AV valve is going to slam shut. And that's to prevent blood from flowing back into the atrium. Remember, when the ventricles contract, we want all that blood to be pumped into the arteries. Now, that slamming shut is actually the first sound that you hear in your heart, sometimes called the lub sound. Now, at this point, the AV valve is closed and the semilunar valve is closed. So imagine now the pressure changes that are happening in the ventricle. You have a closed space and you have a huge push from the ventricular muscles. So all of those cardiac muscles are now pushing blood in a closed space. So we get a spike in pressure in a very short amount of time. So this is what causes this spike to occur. You have a closed system where this valve is closed, and blood has nowhere to go. So pressure spikes. Now, the moment that ventricular pressure gets larger than the pressure within the arteries, or in this case, the aorta, that's what's going to cause the semilunar valves to open. And this allows blood to flow into the pulmonary arteries and into the aorta. Blood is then going to flow into the arteries, and that's going to cause arterial pressure to increase. Because as more and more blood enter the arteries in the aorta, that pressure is going to start to increase. Now, right around the peak of this pressure, this is what's called the systolic blood pressure. This would be the maximum blood pressure that you would hit, would be right around here, right around the peak of aortic blood pressure. Healthy adults may have a blood pressure at around 120 millimeters of mercury, and that's what I represented here on this graph. Now, the reason that aortic blood pressure is going to start to decline is because it's not contained in the arteries, it's flowing out. In the aorta, it's flowing to the rest of the body. So the blood is not staying there, it's moving out. So you get a spike in blood pressure, and then it's going to start to decline. Why does ventricular pressure start to decline? Well, they stop contracting. So once the signal has propagated around the Purkinje fibers, they contracted, and then they start relaxing. As they start relaxing and blood has left the ventricles, ventricular pressure starts to drop. Now, the moment ventricular pressure drops once again below aortic pressure, now the semilunar valves are going to slam shut. And that's to prevent any backflow from blood in the aorta back into the ventricle. That second closure is actually the second heart sound, and that's sometimes referred to as the dub. So you have slamming shut lub and slamming shut dub. So the closing of the AV valves and the closing of the semilunar valves. So now pressure is going to rapidly drop within the ventricles because the ventricles are now in diastole, they're not contracting anymore. And blood has relatively left that chamber. Pressure within the ventricles are going to drop significantly over time. And then once ventricular pressure has dropped below atrial pressure once again, that's going to cause the AV valves to once again open. So now the AV valves are open. This is once again going to allow ventricles to passively fill and the atria to passively fill. And the reason I'm saying passively is because notice that both the atrium and ventricle are in diastole. That means at this stage, neither the atrium or the ventricles are contracting. So you get passive filling, and that blood is coming into through the veins. Now, if this was the point at the highest pressure, what was the point of the lowest aortic pressure? Well, that would be right around this stage. as blood has left the aorta, it's going to continue to decline, and at its lowest point here, this is what's called the diastolic blood pressure. So in this case, on this graph, it would be roughly 120/80. And this is usually how blood pressure is presented. They'll say your blood pressure is 120/80 or 110/70 if you have lower blood pressure. If you have hypertension and maybe high blood pressure, your blood pressure might be 140/100 or something like that. And these are the numbers that they're keeping track of. Generally speaking, it's ideal to have a slightly lower blood pressure so that there's less strain on your arteries, as something that we discussed with cardiovascular disease. This would lower your risk factors for cardiovascular disease because there's less strain on your coronary arteries. A way of remembering blood flow through valves is to really try to figure out which way is the valve allowing flow through. So the atrioventricular valves, it's in the name, want blood to flow from atrium to ventricle. So they're going to be open when the pressure in the atrium is greater than the pressure in the ventricles. They're going to close when the pressure in the ventricles is higher than the atrium, preventing backflow. Semilunar valves want blood to flow from the ventricles to the arteries. So they're going to be open when the pressure in the ventricles are greater than in the arteries, and they're going to close when the pressure in the arteries is greater than in the ventricles. So always think first, which direction do I want the valves to allow blood flow in? That's when they're going to be open. And when they want to be closed is when the pressure where they're going is higher than when it's coming from. And that will, so all these pressure differentials really allow only one-way flow through the heart. These other little bumps here are not necessary for you to understand, but I did outline them in my notes. For instance, if you were wondering why there's a secondary bump in the pressure in the atrium, it's because as these close, some of the blood inside the ventricles will slosh back and hit the actual valves back, and that's causing this slight bump over here. Something similar happens over here when the semilunar valves close, and it causes blood to actually come backwards and hit the semilunar valves, causing this slight little spike. So a summary of this whole process right here is really just understanding how blood is flowing from one area to the next, and the real key way that all of this works is because of pressure differentials. So far what we've talked about in this topic has been all to do with the structure of the heart, the mammalian heart, and how blood actually flows through it. And every time that we've discussed the transport system with respect to animals, we've said, well, the arteries deliver blood to the tissues, and then the capillaries exchange materials. But we've never really explored what's going on when we're exchanging materials. How are capillaries actually exchanging materials, with the actual cells, like what's going on there. So that's what this is all about. This section is all to do with what's actually happening at the exchange zone. So materials are going into the capillaries, but how do we go from capillaries, which are composed of cells, to other cells? Are they touching each other? Well, not really. There's actually another fluid in organisms that I think we often forget about, and that is tissue fluid or interstitial fluid, meaning the fluid in between tissues. Here's an overview as to what's going on. Blood is flowing from the arteries into smaller and smaller arterioles, and then in the arterioles, they're then going into very small vessels called capillaries. And we said the capillaries are almost in this bed form, like we've looked at in B3.1, where they're highly, highly branched in order to maximize surface area. So this is where exchange is happening. Now notice that these are not physically touching the cells, they're exchanging materials in this space in between. That space is the tissue fluid. How is tissue fluid actually formed? So remember, we're going to go from artery to arterial to capillary, and then things are going to move out of the capillaries into the tissue fluid. A concept that will really help you here is the subtopic on water potential D2.3, because that basically will let you know in what relative direction will things move, right? If you recall, what is the formula for water potential? Water potential equals solute potential plus pressure potential. However, solute potential and pressure potential do not contribute to water potential similarly, right? Pressure potential and water potential are positively related. So as you increase pressure potential, that will also increase water potential. But solute potential is inversely related. As you increase solute potential, you decrease water potential. Keep that at the back of your mind when we're talking about this. Because if something is moving in one direction, that you'll have to know, okay, that means that this direction must have higher water potential than this direction. Why has this part have a higher water potential than this part? In this case, tissue fluid is formed from the capillary into the interstitial fluid. This is all formed through something called pressure filtration. We're going to form tissue fluid via pressure filtration, where basically fluid is going to be pressured out of the capillaries into here. So how is that possible? This is coming from the arteriole. That has very high pressure. So, and what did we just say? We said that increase in pressure potential will increase water potential. Therefore, that means that the fluid within the capillaries have a higher pressure potential, so that is going to actually force out materials out of the capillary. So what kind of materials can actually be forced out? Well, as we discussed in this topic in SL, capillaries have a number of adaptations. They have pores or fenestrations, which means that there's actual gaps between these cells. So this allows small materials to be pressure filtered out, forming that tissue fluid. What kind of things are actually leaving? Well, very small gases, something like oxygen gas. We're going to have sugars, things like glucose. We're going to have water, of course. Plasma is mainly water, so that's going to come out. And we're going to have things like amino acids and maybe even some lipids and stuff like that. So what kind of things would actually remain in here? Well, that's going to be things that are too large to actually pass through this gap or these pores. And remember, another adaptation that we looked at in SL was the adaptations for capillaries, in that they have a basement membrane. That basement membrane prevents very large things, like even if they were to be able to fit, like proteins or cells, to actually pass through. So the stuff that's remaining in the capillaries would be erythrocytes, leukocytes or white blood cells, larger proteins. These are going to remain, so these are not being exchanged. But what is being exchanged are all these other smaller things that's entering the tissue fluid. So, okay, now all of this stuff is in the tissue fluid. Now, from the tissue fluid, they're going to be taken up by the cells of that tissue. Depending on the properties, the mechanism in which they're going to be taken up is going to differ. So, if I want to take in oxygen, that is, of course, as we've discussed, through simple diffusion. And because the concentration of oxygen is going to be higher here than in the cells, that's going to flow in no problem. Now, water, as we've already discussed, is going to flow in via osmosis. We have a high water potential here, low water potential in here. And as we've discussed previously, while water can pass directly into cells via the membrane, the vast majority of water entering cells is usually through aquaporins, but it is still osmosis. Now, these other two mechanisms are one that we're going to discuss a lot, and that is co-transport. So in the membrane transport subtopic, we've discussed also this method of secondary active transport. Can you tell me what is the difference between primary active transport and secondary active transport? So primary active transport is all to do with the direct use of ATP in order to move a substance against its concentration gradient. However, this is not what's going on here. This is something called co-transport or sodium co-transport, in which case the sodium concentration is going to be very high outside of cells, and using the potential energy of that gradient, sodium is going to naturally want to go down its gradient into the cell, and effectively glucose or amino acids, depending on the protein, are kind of along for the ride, and they're allowed to be transported against their gradient. So the sodium is going to naturally want to go down its gradient, but then the cell later is going to pump it right back out to keep that gradient high. So now we've moved in this section here, we've had oxygen. Basically, it was really high in the plasma. That then moved into the tissue fluid by a pressure filtration and then absorbed by a simple diffusion. We then had glucose, again, through pressure filtration into the tissue fluid, and that was absorbed by the cells via sodium cotransport. Amino acids, same exact thing. And what's happening is these blood cells and proteins, this stuff is just kind of too big. That's not going to fit through. So if exchange refers to one thing giving something and then taking something in return. So we've effectively now delivered oxygen, nutrients, water, things that the cells would need for metabolism, for growth. So what are the cells providing in return? Well, that would be metabolic wastes. So the metabolic waste that these cells would be providing would be carbon dioxide from aerobic cellular respiration and urea from the breakdown of nucleic acids or proteins and stuff like that. So now, how is the tissue fluid being reabsorbed back into the capillaries, right? If it was only all the tissue fluid was leaving the capillaries, well then we're losing volume out of our blood all the time, and then that would pool in certain areas. We don't want that. So what's actually causing most of that tissue fluid to be reabsorbed back into the capillaries and then leading into venules and then into veins? Let's think back to water potential. We want water and all of this stuff to move from tissue fluid back into the capillaries and then into the venules. So again, how are we going to do this? How can we reduce water potential? In this stage, the net movement was from capillaries into the tissue fluid, right? That implies that the water potential anywhere here in the capillaries was higher than in the tissue fluid. We said that this is largely to do with a really high pressure coming from the arteriole here, right? But now, suddenly we have the reverse. Why would the tissue fluid net move back into the capillaries? Because remember, we're not using proteins to do this. It all has to pass through those pores and through the basement membrane. So what's causing action now to happen in the reverse direction? Well, one, we can use pressure again because now it's the reverse idea. Venules and veins have very low pressure. So the pressure relatively in the tissue fluid will be higher than in the capillaries. So pressure potential is being used as well. However, there's another factor to consider. What happened to the solute potential within the capillaries? Well, we've lost all those other stuff in the plasma, but we've kept all of the proteins. We've kept all of the cells. So its solute potential is actually higher inside the capillaries than in the tissue fluids. So that's going to cause that fluid to follow the area of high solute potential or low water potential, however you want to look at it, to rejoin back into the capillaries. So for this reason, you could say that the plasma within the capillaries has a lower water potential than the tissue fluid. So that means that there's going to be a net movement back into the capillaries. So that stuff that their cells are providing, well, cells would be getting rid of urea, they would be getting rid of carbon dioxide, there's also going to be water, and again, water is going to flow back into. So it's not just that the capillaries want to reabsorb urea and carbon dioxide, they also need to take most of that water back as well, because again, we don't want most of the water to be in the tissue fluids, we want most of the water to be in the plasma. So this is going to rejoin back, and then that's going to rejoin into a venule, and then a vein, and then everything goes back again. So it's almost like the tissue fluid is like the exchange zone. At first, we have pressure filtration that's moving stuff out of the capillaries into the tissue fluid, and then near the end of the bed, we have that being reabsorbed back into here. But is all of the water being reabsorbed? Surely there's still a lot of water and stuff that's still in the tissue fluid. And couldn't that build up over time? If it does, we have a problem, and that's called an edema. If you have a tissue fluid buildup, That's a problem, and we don't want that to occur. So we actually have another whole system of vessels that you may not have realized we have, called the lymphatic system. So the lymphatic system, in addition to capillaries here, you have another whole system of vessels that actually are intertwined within your capillary beds. And their major job is to reabsorb that tissue fluid back. So they will be able to take this back in. Once tissue fluid moves into these vessels, we call that lymph. Lymph is the name of the fluid that's found in here. So I'm doing a zoom in to right here. So this would be tissue fluid that did not enter the capillaries, that's remained there. So the lymphatic vessels are able to take in more of that. Any kind of leftover, that's now entering this section. And if you notice, it has a lot of very similar structural adaptations that capillaries do, which is it has these pores, it has these gaps, and that allows tissue fluid to just naturally go into here, no problem. So if you ever look at a capillary bed, it's not just blood vessels, you also see lymphatic vessels intertwined here. Now, let's consider how fluid might move differently in a lymphatic vessel. The lymphatic vessel is not connected to the heart. So, how do we move something? Let's think back. Question. How can we help move fluid through a tube when either we lack a pump or the pumping pressure is very, very low? Similarly to veins, because the fluid moving through lymphatic vessels is at extremely low pressure, we need a way of moving it through, and they're even going to be even lower pressure than veins because they're not even connected to the heart. So how are we going to move a bunch of fluid through when we don't have a pumping action? Well, we can't really use transpiration and capillary action because the vessels are going to be fairly large, so it's not going to work. But we can use the same adaptations that a lot of veins have in, let's say, the legs, which is they have valves, which allow only one-way flow through the lymphatic vessels, but the actual walls are actually quite thin. And that's because they can be surrounded by skeletal muscles, and as the skeletal muscles contract, it literally pumps the lymphatic fluid up, similarly to how your calf muscles could actually flex to cause blood plasma to move up your veins and up your leg. Now, because this is again not connected to the heart, lymph is moving very, very slowly, like something like 100 to 500 times slower than that in veins. So the movement is very, very slow through lymphatic, but it is still moving. And again, it's largely due to these major adaptations. So we have lymph now. Does that really solve the problem? We've net lost water right now from the capillaries into tissue fluid, and then that tissue fluid entered the lymph. I want it to ultimately return back into the blood. So what's causing it to return back into the blood? Well, the lymphatic system actually will rejoin and deliver it back into the veins. So all that tissue fluid that it took in as lymph will then be returned back into the vein. However, there's a few stops before it does that. It won't return it directly first. It will actually pass through these nodes called lymph nodes, which in fact you can probably feel. If you feel under your armpits, you feel little bumps. If you feel here, you feel little bumps. Those are lymph nodes. So what exactly is a lymph node? Well, a lymph node are little small organs throughout your lymphatic system. In fact, you have many lymph nodes throughout your system. So what's the point of them? Well, notice you have a bunch of different lymphatic vessels all feeding into this one node. These would be called afferent lymphatic vessels, meaning coming in, and this would be an efferent lymphatic vessel. So all of this stuff is coming in here. This actually kind of acts as a filter, because let's say some of that stuff that escaped your capillaries were bacteria, were viruses, were things that really are foreign to the body. Well, that's going to be collected by the lymph, and then before returning it back to circulation, it has to almost pass a security checkpoint. And that's what these lymph nodes are. Because what else happens at lymph nodes? This is where white blood cells mature. This is where most of your white blood cells are maturing and are being held. So it's almost like before returning back to circulation, you have to pass through a security checkpoint. That security checkpoint being lymph nodes. So now that tissue fluid is coming in here, and anything that shouldn't be there, white blood cells are right there, and they hopefully will destroy it. And in fact, you'll likely know if you're undergoing a pretty serious infections because your lymph nodes may be swollen. In fact, if you go to the doctor and they're like, hey, your lymph nodes are swollen, that's because there's likely kind of a battle happening here because there may be an infection going on. So that's kind of an early sign. Your doctor may actually feel right here and they'll say, actually, your lymph nodes are swollen. That could be a sign that you're currently undergoing an infection. And this is a concept that we'll look at more when we do defense against disease in another subtopic. So your lymph nodes and your lymph system is actually really, really awesome. And I think kind of no one knows it exists. It's like a very useful system because one, it takes back any of that fluid that you lost into the tissue fluid back into circulation. But before doing that, it acts as a filter for any foreign substances that should not be there. The whole point of this part right here was what's actually happening at the capillaries? How exactly are we getting a net flow first from capillaries to tissue fluids, exchange in the cells, and then tissue fluid back into the capillaries? And then the lymphatic system is addressing the problem of, well, how are we ultimately getting that tissue fluid back into circulation? And the added bonus of let's try to filter it out of any foreign substances that shouldn't be there. Okay, so similarly to standard level, where we looked at both transport at the animal and plant level, we're going to do the same thing with higher level. So we're now going to shift gears into plants. So we've already explored how water moves through plants, and that would be through the xylem, through the process of transpiration. But what we're going to be looking into here is how exactly do sugars and other substances move through plants? Through this phloem and through a process called translocation. So if we recall, here's a cross section of a root of a dicot. And then right in the center here in this cross is we have xylem. And then from the inside out, remember the outermost layer would be the epidermis, and then you have the cortex, and that will lead into here. So let's first ask the question, how exactly is water entering the plant in the first place? I've talked about this briefly in standard level. This is now an official learning outcome in higher level. And once again, our knowledge of water potential is really, really key here. Understanding water potential will allow you to understand the direction of flow, just like we just looked at with respect to capillaries and fluid tissue. That all had to do with water potential. So let's look at what's going on in the roots. So let's zoom in to this section right here. And let's take a look at a single root hair cell. So a root hair cell, again, predict why a root hair cell might look like this. So a recurring theme that we've explored in B3.1 was if anywhere where I have a lot of exchange or transport, I want to increase surface area to volume ratio. And in this case, the roots would want to maximize surface area to volume ratio. So these cells would be those right on the epidermis facing out. So they have these projections coming out, massively increasing surface area to volume ratio. And as we explored way back in A1.1, we said that water can actually move through soil because soil is not fully packed together. So we have water all surrounding the soil. And again, we want water to flow net from the soil into the root. Now, roots are also going to have plenty of water. So how exactly do we consistently ensure that water is moving in? So we're going to have to decrease the water potential in the roots. And to do that, they're actually going to pump in ions. So the root hairs are actually going to use active transport. So they're going to use ATP to transport ions that I've shown here in just these red and orange dots. It's not important that you know which ions they are. And they're going to move them against their concentration gradient. Anytime that I'm doing active transport, I'm trying to build a gradient. So if I want to build a gradient, I'm going to use active transport to do that. And so that's going to cause the solute potential inside of the root to be very, very high. As you increase solute potential, you decrease water potential. That's going to lead to water now flowing in, right? Water is now flowing in. Fantastic. Now there are multiple ways now that we have to move water from the epidermis into the cortex, into the xylem. There's two different pathways that can be taken. One pathway is called the apoplast pathway. One pathway is called the symplast pathway. Water can actually move through the cell walls in something called the apoplast pathway. So what I've shown here in purple is actually that intercellular space is actually being able to flow from cell to cell to cell in the cortex. So it's actually flowing through the cell walls from one cell to the next. And the other major pathway that's taken is something called the symplast pathway. So that would be from cytoplasm to cytoplasm. Now, how is that possible? We have cell walls and a plasma membrane. What's doing that? Well, there's actually gaps. These gaps are called plasmodesmata. So these gaps allow water to flow from the cytoplasm of one cell in the cortex to the next, to the next, to the next, all the way through. So far, we've flowed water in to the epidermis through the root hair cells. Now through the apoplast and symplast pathway, we're moving from cortex cell to cortex cell to cortex cell. Now, we kind of want to funnel all that water to the xylem. We don't want it to spread out everywhere. So there's another adaptation here, something called the Casparian strip. We want to funnel water from the cortex and ensure that it's entering into the xylem. So at certain points, when we get to the cortex cells that are very near the vascular bundle in the center of the root, we have this layer called the Casparian strip. The Casparian strip is highly, highly impermeable. So where we were getting all of these pathways that could actually move from cell wall to cell wall and maybe from plasmodesmata to the next, this is basically like, no, you can't do that anymore. And it's basically preventing, it's forcing all of that water into this cortex cell, and the only where it has to go is the xylem. So it's funneled into the xylem this way. So the Casparian strip is actually quite near the vascular bundle to ensure that water is funneled into the xylem ultimately. So now, again, you have to ask yourself the question, if water is moving from the cortex into the xylem, why is it doing that? That means that the water potential has to be higher here than it is here. So why? What would cause the water potential to be higher in the cortex than in the xylem? So what you see here is similarly to what we did in the root hair cells, we're using active transport to pump ions from the cortex into the xylem. And the water potential has now also decreased, because if I'm increasing solute potential, I'm decreasing water potential, and now it's going to enter in this way. As I'm increasing a bunch of water, that's going to increase pressure potential. So the pressure potential now here is going to be high, and that's going to force water up. So we said before in transpiration, what's causing water to move from roots to shoots? Well, we said it was largely driven by low pressure potential in the leaves compared to the roots, which had a high pressure potential. And that's true. As long as water is evaporating and leaving out of the stomata, that will continue to occur. But this also will contribute to it to ensure that pressure is really high in the roots compared to the shoots. So it's actually both of these factors combined. And actually this does allow some degree of movement even if transpiration rates are low, or even if the amount of evaporation in leaves are low. You're still going to get high pressure in the roots because of this system. So this root pressure potential is really, really key. If you look at the first learning outcome, it says generating root pressure in xylem vessels via active transport of mineral ions. So bring all of that together. By actively transporting ions from the cortex into the xylem. I've increased solute potential, decreased water potential, causes an influx of water that increases hydrostatic pressure, and thus water is going to get more driven up. So it's kind of a combination of both the low pressure in the leaves and the high pressure in the roots, allowing this process to occur. We're finally going to talk about the phloem here, and we're going to use something called the pressure flow hypothesis for translocation. So this is a mechanism that has been proposed to explain how sap or sugar water or other kind of solutes within water are transported through the plant. And they're primarily transported not in the xylem, but in the phloem. So let's discuss step by step how we're actually getting sugars and other vital metabolic nutrients to other parts of the plant, right? Because if all you knew was transpiration, you may be thinking that plants are kind of cooked, because transpiration is unidirectional. It's only going from roots to leaves. So roots need sugars. All the sugars are being made in the leaves. All the proteins are being made in the leaves. So they're like, when am I going to get anything? I'm sending you all the water up. How are they ever going to get any nutrients? So translocation is actually bidirectional. It allows flow kind of anywhere where the plant needs it. But how can we do that? And again, a recurring theme that I know I'm repeating myself here, but a recurring theme is if I want fluid to move in a direction, I'm going to use pressure potentials. I'm going to use a pressure differential from one area to another area. So this is yet another theme that we're going to be encountering here. So let's begin where all translocation would begin, which is something called a source cell. Because remember, translocation is the movement of these metabolites, things like sugars, proteins, whatever. So where are they primarily being made? In the source cells, or areas that are effectively doing high rates of photosynthesis. So these would be largely the leaves. Palisade mesophyll cells, right? These would be undergoing high rates of photosynthesis. So they're going to be producing a lot of glucose. That glucose is ultimately going to be in their cytoplasm. They may store some of it, remember, inside of their chloroplast as starch, but they're going to have a lot of glucose here. We are actually going to use active transport to move glucose into this cell called a companion cell. So this is a cell that is connected to this tube. So the phloem comprises two elements here, something called the sieve tube element, or sieve tube cells, and the companion cells. Now we've explored this in the cell topic when I said that these cells are kind of a weird exception to cell theory. They're atypical examples of cell theory. The sieve tube cells are anucleated. There's no nucleus here. They have very little cell components, but unlike the xylem, they're alive. So how? Well, they kind of have a life support system. That life support system is the companion cell. The companion cell is kind of doing most of the metabolic activities for the sieve tube cell. So we're producing a lot of sugars and metabolites inside of the source cell. We are now going to actively transport that glucose, or sucrose really. And I should mention this as well. Plants don't actually transport around their bodies glucose. They actually transport it primarily as sucrose. Inside the source cells, they're going to almost package glucose as sucrose, as a disaccharide. That disaccharide then gets actively transported into the companion cell. And this is also done using a co-transporter. And in this case is something called the proton sucrose co-transporter. Now, without me explaining anything, tell me how this would work. I'm moving sucrose from here to here using a proton sucrose co-transporter. What's going on? It means that the companion cell is actually actively moving protons out. It means the protons are higher outside the companion cell than inside. Protons are going to naturally want to flow down due to their concentration gradient. So we have potential energy here. And then sucrose is kind of, again, along for the ride, moving into the companion cell. So now we've created a very high concentration of sucrose within the companion cells. Now, this is going to passively flow into the sieve tube cells through the same structure that we've already talked about, plasmodesmata. So there's also gaps here, and they're going to flow constantly from the companion cell into the sieve tube cell. If you were wondering why active transport had to happen here in the first place, this is why. By having a really high concentration of sucrose here, this is passively always going to flow into the sieve tube cell constantly. So they're creating, as it's being made, sending it out, sending it out, sending it out, sending it out, sending it out. High concentration of sucrose here, and then it's going to passively enter here. So let's look at that now. If I'm constantly adding in sucrose from companion into sieve tube cell, what's happening to the water potential? If I am constantly getting sucrose diffusing into the sieve tube cells, this increases solute potential, decreases water potential, and what's right next to phloem? Xylem. Xylem now, which is a big tube of water, is going to flow from an area of high water potential to low water potential, and water is going to flow from the xylem into the phloem. So this is why, hopefully now it's starting to make sense, why plants bundle their xylem and phloem together. So xylem are almost acting as the main highway of water, but it's really what's causing the push to happen in the phloem. So now, if I have an influx of water into the phloem, what's that going to do to hydrostatic pressure? It's going to increase. Hydrostatic pressure, now that the hydrostatic pressure is high here, it's going to flow anywhere where hydrostatic pressure is lower, including up or down. So this is why translocation can actually be bidirectional. I can move sap anywhere where the hydrostatic pressure is lower. So we describe transpiration as a flow from roots to shoots. We would describe translocation as a flow from source to what are called sinks, or anywhere that need these metabolites. So we have a high area of hydrostatic pressure here. Now my diagram shows this flowing down just to make a point that you can flow it down, but this could also go up, sideways, anywhere, right? So area of high hydrostatic pressure here, it's now going to flow down the sieve tube cells in a process called bulk flow. So bulk flow really just describes sap flowing from an area of high hydrostatic pressure to low hydrostatic pressure. So that's really what's going on here. Then we finally made it to where it's needed, something called a sink cell. And again, a sink cell can be anything that needs growth. So this could be a flower bud, this could be a shoot, this could be a root, anywhere where there's a lot of growth and therefore they need a lot of nutrients for metabolism, for growth. So don't think that the sink cell is only going to be roots. It really can be anywhere that needs these sugars, right? So look at this process. We're now going to do the reverse. So because we've transported all of these solutes here, these are going to go down their concentration gradient from inside the phloem into the companion cell. So sucrose is going to move from the sieve tube cell through plasmodesmata into the companion cell. Now, to ensure that it's always moving in this direction, we're going to do active transport, but now into the sink cell, to ensure that the companion cell always has lower concentration than the sieve tube cell. So it's almost like the reverse process that we just saw was happening at the source. So if, therefore, the sink has a lower sucrose concentration, then sucrose is going to passively diffuse in, no problem. And if that sucrose concentration is actually higher, they can also use active transport. So we're constantly making sure that sucrose is entering the cells that need it, or these sink cells right here. Now, again, we kind of have a similar dilemma that we had with the tissue fluid, which is I want to make sure that this fluid doesn't stay in the phloem. I also need to return some of it back to the xylem. Well, once again, water potential is going to be our friend here. As sucrose is leaving the phloem, that's decreasing solute potential, increasing water potential. Therefore, water will naturally flow from the phloem sieve tube cell into the xylem and enter recirculation all over again. So you can see that transpiration and translocation are linked. You really are not going to get much translocation happening without transpiration because you rely on the flow of water through the xylem. So both of these processes are highly, highly linked and related to each other. Now, just like we did when we looked at transpiration, we discussed what are some adaptations that xylem have that allow this to happen. Let's do the same thing for phloem. So what adaptations do these cells have that allow translocation to occur? Well, think about all of the active transport that was required here. The companion cells are doing a bunch of active transport. Remember, they're kind of like their life support system, so they're going to have enormous ATP demand, so it's probably unsurprising to you that they are packed full of mitochondria. They have a very high density of mitochondria within their cells to allow for very high rates of aerobic cellular respiration. The next thing, as we've already discussed, these gaps are really, really important that allow an easy passive flow from high concentration to low concentration, those being plasmodesmata. So they're basically just small channels that connect adjacent cells with each other. And they allow very, very, very rapid movement. And again, this is not just for sucrose. Consider where companion cells are related to phloem cells. All of metabolic activity has to be done by here. So as they're creating ATP or whatever, that can easily be moved just through diffusion very, very quickly through plasmodesmata. So it allows a very fast connection kind of between the life support system and the tube itself, right? The third adaptation would be, well, we said an adaptation that xylem had that allowed them to flow very, very easily is that the cells were dead and they effectively fused together and there were no partitions between cells. But these phloem tube cells are actually alive, so you can't remove partitions completely because they still would have to retain their own shape. So how do I allow movement through a tube without killing the cells and doing something like a xylem? Well, they have these plates that are perforated. So you have these perforated plates joining one cell to an adjacent sieve tube cell to the other. So these sieve tube plates are basically perforated partitions between one cell and another, allowing rapid movement through. And lastly, kind of the same adaptations that xylem had, this is ultimately a tube. So I want to reduce the amount of stuff in the way. So there's no nucleus, there's a vastly reduced cytoplasm, and there's a vastly reduced amount of organelles within sieve tube cells. So this again vastly promotes how efficiently I can transport sap through these cells. So overall, this whole section was discussing how things can move through plants. And I know a lot of people may be slightly more bored by plants than they are by animals, but I think the system really is very fascinating because it's maybe answered questions that you've never even had, which is you probably never considered how things move all over plants that don't have a heart, that don't have a pump. How are they actually doing this? And through vascular plants, it's kind of a lot of similar systems. Actually, a lot of similarities between how they transport things and how we transport things. And kind of if there's one underlying mechanism that hopefully ties all of this together that you're seeing, it's pressure. It's water potential and pressure. This is like a constant recurring theme when we're talking about transport. If I want to move some fluid from here to here, I'm going to manipulate pressure, I'm going to manipulate water potential. And that's kind of tying all of this together. Let's go over the additional higher level learning outcomes for subtopic B3.2 Transport. Similarly to subtopic B3.1 Gas Exchange, some of the learning outcomes deal with the processes occurring in animals, and some of the learning outcomes deal with the processes occurring in plants. So these first two learning outcomes really had to do with the exchange occurring between capillaries, the tissue fluid, and the actual cells within different tissues, and how all of this is being driven by differences in water potential and in pressure. This learning outcome deals with the role of the lymphatic system in collecting excess fluid and returning it back into circulation, and also acting as almost like a check-in point and a barrier for any potential pathogens that have leaked out. With respect to circulation, we looked at the differences between single circulation in stuff like bony fish and double circulation in animals such as mammals, and how these two systems actually differ. We then did a deep dive into the actual structure of the mammalian heart, and we looked at how the different anatomical features are adapted to their function in allowing one-way flow of blood through the heart. We then explored the different stages in the cardiac cycle, both with respect to systole or contraction and diastole with respect to relaxation. And then finally, with respect to the plants, we explored first how pressure in the roots is generated via active transport and how water can actually flow from the epidermis through the cortex and then ultimately into the xylem, generating pressure flowing upwards. And then finally, the process of translocation and the role that the companion cells and the sieve tube cells have in actually moving sap to areas of high need or sink areas. If you wanted to learn further, which I always encourage you do, this last page is a good place to start. Here are books in which I use in order to create these resources and the relevant page numbers in mind if you wanted to check them out. There are many 3D models that I've recommended here for really exploring the cardiovascular system and the heart in 3D and high resolution that really allows for more understanding than just looking at a 2D image. And this is also a fairly good animation as to translocation in plants. Simulators and interactives are a great way to really start to understand, again, quite complex processes. And some simulations which I've already used in this lesson were that of the circulatorium, or even in the cardiac cycle, showing how the heart is actually beating at different stages. If you want to practice labeling, here is a really good spot. And if you want to do a little bit extra and actually go into the role of a doctor and explore cardiology, this is a really nice activity. Finally, these articles are ones that I would really explore for just further reading because I think they're really, really fascinating. For instance, this has to do with the future heart transplantation and certain advances that have come about recently. And these explore cardiovascular disease and really looking at risk factors with respect to coronary heart disease. Lastly, I would always encourage that students not just learn for the sake of a test, but learn for the sake of learning, learn for enjoyment.